attendance. I know there will be latecomers that I can't anymore record their attendance while I am discussing. Okay, so good morning again. This will be the first lesson for Q3, and throughout or the entire Q3 will be focused on genetic engineering, which is actually the thrust in modern biotech. Okay, genetic engineering is actually modern biotechnology. So for Module 1, we'll be discussing the tools of genetic engineering. I know for sure that you have already read the module that I have uploaded a few days ago. Okay, so I think you already have an idea of the tools that are used in genetic engineering, in biotechnology, or in modern biotechnology. Okay, so okay, so we'll try to or just read. Everybody please read the introduction of the topic. So 1, 2, 3, go. You can turn on the microphone. Hello.
Hello, congratulations for making it to this quarter.
Congratulations for making
Are ready for another round
Ready
of
for another round of
Have you already
watched
The movie X-Men, the science
science
fiction movie that depicts individuals with enhanced genetic
modification
modification that give them special
ability?
abilities?
This
Module,
we will be
introduced
introduced
to the basic
concepts
concepts of genetic engineering.
This
Module
also
also discusses different tools commonly used in genetic engineering.
Okay, thank you for that. Thank you. So in this module, again, you will be introduced to basic concepts, only the basic concepts about genetic engineering, because the advanced concepts, I think, will be discussed in when you go to, or when you enroll STEM in senior high, or when you enroll science or biology related course in college. Okay, so what we will tackle today will only be the basic concepts of genetic engineering. Now, after going through this module, you are expected to describe the different tools used in genetic engineering. So for the entire lesson, we will be guided with these objectives. First is to explain genetic engineering. So I hope that after discussing the topic, you can explain to yourself, you can explain to your friend what genetic engineering is. Another objective is to know the history surrounding the progress of genetic engineering. Another is to identify the tools commonly used in genetic engineering, which is actually the highlight of today's discussion. And the last one is appreciate the importance of genetic engineering tools. Okay, so again, we will be guided with these objectives throughout the lesson. Okay, so if you still remember the last discussion that we had in quarter 2, I think the last topic was on mutation. Mutation. So in first place, what is mutation? Again, you still remember the definition of mutation, or how will you define simply mutation? Anybody from the class? Any volunteer? Any volunteer? Can I ask somebody from Mango class? Yes, Pable, Margaret. Okay, so what is mutation? So what have you learned in mutation? Okay, yes.
It is a change that occurs in our DNA sequence either due to mistakes when the DNA is copied or as the result of environmental factors such as UV light, cigarette smoke, and
Okay, so thank you for that. Zoe. So mutation is actually any change in the DNA. Any change in the DNA will cause mutation. So any change in the DNA, even just a very slight change in the DNA will cause mutation. Okay, so let us first answer as a sort of review. We will answer the 8 questions here. You will refer your answers to the word pool in the module. Okay, so let's take the first one. Number 1, a mutation that occurs in gametes. So what is that? A mutation that occurs in gametes. So anybody can answer.
Germline mutation. Germline mutation.
Very good. Germline mutation. So any mutation that occurs either in the sperm cell or in the nucleus or the DNA of the sperm cell or the DNA of the egg cell is collectively called as germline mutation. And number 2, a mutation that occurs in other cells of the body. Anybody?
Somatic mutation.
Very good. Somatic mutation. So a mutation or a change in the DNA of skin cells, a change in the DNA of liver cells, a change in the DNA of cardiac cells and other cells of the body is called as somatic mutation. So mutation that occurs other than egg and sperm is called as somatic mutation. We'll go to number 3. Mutations that change chromosome structure.
Chromosomal
Alteration.
alteration.
Okay, so the answer is correct. Chromosomal alteration. So a change in the structure of the chromosome, which is actually more dangerous than genetic mutation. It is more dangerous than genetic mutation. So chromosomal alteration or chromosomal mutation. So in chromosomal mutation, there is a change now in the structure of the chromosome itself. Kung nahalaba, nahalipot, actually the main cause for that is breaking the arm of the chromosome. Okay, so number 4, a change in a single nucleotide in DNA. So
Point mutation.
okay, very good. Point mutation. Point mutation. Even just a single nucleotide change in the DNA will cause mutation, or it will make a difference in the individual. Okay, we go to number 5. a deletion or insertion of one or more nucleotides that
Frame
changes
shift
the
mutation.
frame, the reading
Frame
frame.
shift
What do you call?
mutation.
Okay,
Frame
so
shift
Frame shift
mutation.
it's called as frame shift mutation, which is more dangerous than point mutation. Okay, so we go to number 6, results to defective protein in connective tissue.
Marfan
syndrome.
Marfan syndrome.
Marfan syndrome is correct. Marfan syndrome is correct. Marfan syndrome is caused by a defective protein in connective tissue. Okay, so another, number 7, abnormal hemoglobin protein in red blood cells. I think I mentioned this last time.
Sickle cell anemia.
Anemia. So we call this mutation as sickle cell anemia, which is actually fatal. So when a newborn child is diagnosed to have a sickle cell anemia, it simply means that there is something missing in there is a missing nucleotide in the DNA of the cell. I think the main cause of this is the type of mutation for sickle cell anemia is point mutation. Point mutation. A single nucleotide affects the protein that is being produced. So number 8, lack of a substance needed for bones to absorb minerals.
Vitamin D
resistant. Vitamin D resistant. Vitamin D is actually needed for the absorption of a certain substance in the bone, in bone cells. So if the bone cell cannot, or if the bone cell resists absorption of vitamin D, then definitely bones will then be affected. Okay, so thank you for correctly answering all the questions. Since you were able to answer all the questions, I'm pretty sure you are all ready for quarter 3, which is genetic engineering, or simply modern biotech. Meron pa, number 9 and 10. Reduced activity of a protein needed for blood clotting, which is connection to number 7. Reduced activity of a protein needed for blood clotting.
Hemophilia.
Hemophilia, very good. A change in DNA?
Mutation.
Okay, so let's take now the definition of genetic engineering, which is actually objective number 1. Okay, so what is genetic engineering? In the previous lessons, you have learned about DNA, a genetic material that carries the information needed to direct protein synthesis and replication. Genetic engineering, sometimes called genetic modification or simply modern biotechnology, is a biotechnological process that helps modify or improve the genetic traits of a particular organism. So this is actually the main highlight of genetic engineering. The purpose or the main objective of genetic engineering is to modify or improve the genetic traits of a particular organism. So this is again the highlight of genetic engineering. Again, to modify or improve the genetic traits of a particular organism. How is this done? It is done by manipulating the genes. So you have to manipulate the genes, meaning to say, genetic engineering starts at the cellular level. So if you want to change an organism, if you want to change your plant, if you want a desired trait for your plant, if you want a desired trait for your pet, iton imo pag-modify, diri na dida mismo iton organism. No, you have to start it at the cellular level. Okay, so again, you will start at the cellular level, meaning to say, i-modify mo dito itong DNA. So once modified, so na-insert mo na ang desired trait that you want your pet to have, the desired characteristic that you want your plant to have. So ma-modify mo na nga ni itong nucleus or the DNA of the nucleus in that cell will undergo mitosis. So pag-divide niya, pag-undergo niya itong mitosis, it will copy the inserted genes, or it will copy the desired genes that you put into the cell. So the results of mitosis will then become, or the cells that were the result of mitosis will then form a new individual, a new organism. And in that organism, you can find now the desired gene that you want for your pet, or the desired characteristic that you want to see in your plant. Again, genetic engineering starts at the cellular level. So you manipulate the genes in the DNA at the cellular level. Diri nga di dayton organism may ma-manipulate mo itong genes itong tanan nga mga cells. No, it will be very, very hard for you to do this. Okay, so genetic engineering again is done by manipulating genes, identifying the desired gene, and transferring it to another organism. Okay, again, how is genetic engineering done? First is manipulate the genes. Then another is identify the desired gene. Then transfer the desired gene into the cell of another organism, or into the DNA of another organism. Okay, I hope you're taking down notes. This is quite important. The product of this process is called a genetically modified organism, or simply GMO. Okay, so once the organism has a foreign gene inserted into it, then you call the organism as genetically modified organism. If it is a plant or a crop, you call the crop as genetically modified crop. Okay, let's say for example, an imo rice plant, ginbutangan mo hin gene that can resist weeds. Ginbutangan mo hin gene that can resist pests. Then your crop will become a genetically modified crop, or a genetically modified rice plant if it is a rice. Okay, kung petsay hiya, then you call the petsay as genetically modified petsay. Okay, or transgenic organism. Transgenic organism. So sumeri nga ni kita transgenic organism, it simply means trans, meaning transfer of gene, transgenic or transgene, transfer of gene. So if a certain gene is transferred to an organism, and that organism manifests the desired gene or the inserted gene, then you call the organism as transgenic organism. Kuha niyo. Okay, so the product of genetic engineering is called as genetically modified organism or genetically modified crop, if it is a crop, and transgenic organism. Okay. Now, genetic engineering can be applied to any organism. Or, in other words, genetic engineering is not only applicable to crops, it is not only applicable to livestock. So, meron nga livestock, ito ng mga carabao, cow, goat, pig, what else? Horses. So they are collectively called as livestock. So genetic engineering is not only applied to livestock. It is not only applied to pechay, to kamote, to gabi, to corn plants, to rice plants, which we collectively call as crops, but to all organisms. Genetic engineering can be applied to any organism, from a virus to any organism, from a virus to a giant organism. For example, in genetic engineering can be used to produce plants that have a higher nutritional value or can tolerate exposure to herbicides. Okay, sigunta, you want your crop, you want your kamote to have another, or you want your kamote to be more nutritious. There are carbohydrates ada. You want to insert iodine, you want to insert vitamin A, you want to insert vitamin C, vitamin or other vitamins in your kamote, you can do it through genetic engineering. Okay? So we go to the history of genetic engineering. Now, genetic engineering didn't happen in just a click of a finger. Genetic engineering didn't happen in just a wink of an eye. So maiha ang pag-studio, genetic engineering, the genetic engineering that we have now. Okay, so it actually started when the DNA was discovered. Okay, so the DNA was discovered in 1953. Through the discovery of the DNA, many things happened. Through the discovery of the DNA, many scientists became so interested in the DNA that they were able to discover many things about the DNA. Okay? They were able to discover many things using the DNA. Okay, so from 1953 to 1918 and even up to now. So the genetic engineering that we have at present is far more advanced than the discovery in the 20th century. The 20th century or the 1900s. Okay? Okay. So, direta na pagtatag usa una kay mag-iiha kita duro ba tanda damang kita pag hinahiton as it is internet. So the first transgenic animal was discovered in 1981. The first transgenic animal. Okay. The first BT corn, do you still remember the BT corn? BT corn? Bacillus thuringiensis corn that is resistant to tests, it was discovered in 1988. Okay. The discovery of the palindromic repeats, which we will discuss later, it was discovered in 1993. The palindromic sequence or the palindromic repeats was discovered in 1993. Dolly the sheep was cloned in 1996. Okay, another, the glowfish. I don't know if you are familiar with this. Children, are you still there?
Yes.
Okay,
Yes
very
po,
good.
ma'am.
Yes, ma'am.
Mawaray to audience. Anyway, the glowfish was discovered in 2003, so 21st century na. Glowfish. Are you familiar with glowfish? Or
No.
anybody who has a glowfish at home? Hinot may ada glowfish kasi kung ano niya na kung baga trend na niya na ito nag-aquarium. The fish that glows in the dark. Trademark it niya is glow, G-L-O, glowfish. An iya hini kuan, an iya hini gene that made the fish glows in the dark was taken from a jellyfish. Diba jellyfish glows under the sea? So an gene that let the jellyfish glows under the sea was transferred into an aquarium fish. Definitely, kami ada naman niya gene to glow. So the fish, as it is, the characteristic for glow was observed in the fish, in the aquarium fish. So the fish then glowed in the dark, which is actually very beautiful to look at when, or in the aquarium when lights are turned off. Okay, so for sale na ini, this is already for sale in the market. So it was discovered in 2003. Stem cells. The pluripotent stem cells was first discovered in 2006. Okay. What else? The human trials for palindromic repeats. CRISPR is an CRISPR, clustered regularly interspaced palindromic repeats, which was applied to humans. It was first applied in 2018. Okay. Scientists did not stop using the DNA in 2018. Until now, they are still they are still using the DNA to discover many things for the improvement of life, to modify and improve organisms, specifically crops and livestock. Okay, so we will continue with the tools in genetic engineering, which is what I have, as what I have said just a while ago, which is our highlight for today's discussion. Okay, so tools. In genetic engineering, we actually use tools to modify a certain cell, or to modify a certain organism. The tools referred to here are not the tools that we often see in the laboratory, the tools that we often see in farms, the tools that we often see in how was this? In carpentry. So they are not the common tools that we see in our environment. They are not the common tools that we use in the kitchen. The number one tool used in genetic engineering are enzymes. Enzymes. You know what an enzyme is? Anybody who knows what an enzyme is? Malex, are you there?
Yeah.
Okay, can we hear from you? So what do you know about enzyme? Or anybody from the class? Anybody from Santal class? Ah, yes. Ambil is raising his hand. Yes, Anna, can we hear from you?
They're used for digestion.
Yes, Hannah.
They are used for digestion.
Enzyme. Okay, thank you for that. Chad is raising his hand. What is your idea about enzyme, Chad?
Parang catalyst with living organism.
Okay, so thank you for that.
Protein that speeds up chemical reactions.
Who answered that? Who answered that? Please identify yourself. Yes, Anna. Okay, so yes, thank you for that. So enzyme is actually a substance, a protein. Enzyme is a protein. Enzymes actually speed up or slow down any chemical reaction. So our cells undergo many chemical reactions. Now, for the cells, for cellular processes to take place, for cellular processes to happen, enzymes play a great role for that. Okay. You still remember replication, the enzymes used in replication, the DNA polymerase, in what else? The DNA ligase. Those are enzymes that are very important for replication to occur. Even in transcription will not occur if there are no enzymes that help do the process. So enzymes in cells are very, very important. In genetic engineering, enzyme is a very important tool in the modification of a certain organism or a certain product. This is very important. And these enzymes, which we call as restriction enzymes, serve as molecular scissors during the process. Remember that cell is very, 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 very small. DNA is found inside the nucleus of the cell. So if cell is small, nucleus is smaller than the cell. And in this very small nucleus is the DNA. And the DNA are the genes. Gumamit ka nga nin scalpel, gumamit ka nga nin gunting, matutuhay ka ba? Class. So if you will use a scalpel, if you will use a pair of scissors to cut a DNA, matutuhay ka ba?
No.
Definitely not. Definitely not. So what the scientists did, they used the enzymes to cut a segment of a DNA molecule, or to cut a part of a DNA molecule. Because they found out, while they were studying the DNA, while they were studying the cell, they found out that there are enzymes that can cut certain sequence of a DNA molecule. And we call these molecular scissors that are able to cut DNA molecules as restriction enzymes. Okay, so restriction enzymes are used to cut DNA into smaller fragments. The cuts are always made at a specific nucleotide sequences. Different restriction enzymes recognize and cut different DNA sequences. Please take note of this. Restriction enzymes cut DNA into smaller fragments at a specific nucleotide sequences. Specific nucleotide sequences. Enzymes work at a specific nucleotide sequences. Enzymes are very specific. They don't cut the DNA just anywhere. Gadiri nga ala, type ko didi pagkat iuot dungkot hiya. Type ko didi tunbot nga igkat. Type ko magkathin 5. No, doesn't happen. Restriction enzymes are very, very specific. They cut at a specific recognition site in the DNA. Again, restriction enzymes cut certain restriction enzymes cut a specific nucleotide sequence in a DNA molecule. They don't cut just anywhere in the DNA. They are very, very specific. Okay, so different restriction enzymes recognize and cut different DNA sequences. It's okay, Ade. Yeah, okay. So the place or the specific site that the restriction enzymes is called as recognition site. Again, di dahil itong na DNA, kundi inhera na utod, we call that specific site as a recognition site. Okay? Its restriction enzyme recognizes a different and a specific recognition site or DNA sequence. Recognition sites are usually only short, from 4 to 8 nucleotides. 4 to 8 nucleotides. Direct haglab ay hindurot eragin kakat. No. 4 to 8 nucleotides. Okay? DNA cut with a restriction enzyme produces many smaller fragments of varying sizes. So once na-cut na nira, may ada makakat nga halipot, nga fragment, or segment, may ada makakat nga halaba, nga segment. Okay, also, it actually depends on the type of restriction enzyme that cuts a DNA molecule. Okay. Wait lang ha, anay ha, madali lang. Okay, I'm back. Okay, so this is how the restriction enzyme cuts a certain segment of a DNA molecule. Let's take, for example, this one. Inihiyang apart okay, so inihiyang apart was cut by a certain restriction enzyme. So gincut niya ining apart ang DNA, and this is now the result. Okay, from 1, the DNA now becomes 2. Hino ang nag-cut? Ano ang ginagamit? It is not an X. Okay, so it is not an X. It is not a pair of scissors. It is not a knife. Dere ginamit hin knife, dere ginamit hin haksa, dere ginamit hin gunting, but restriction enzyme. Okay. So in the module, there are 2 types of restriction enzymes. One is exonucleases, and the other one is endonucleases. Exo and endo. So exonucleases catalyzes hydrolysis of terminal, dahil mga katapusan itong DNA molecule. So asat iya ginuotod, the endonucleases, terminal nucleotides from the end of a DNA or RNA molecule, either 5' to 3' direction or 3' to 5' direction. So we have exonuclease 1, exonuclease 2, and others. Okay. So again, when we say exonucleases, this is the type of restriction enzymes that cut at the terminal nucleotides. Terminal, dito mga katapusan, dito mga end, dito DNA molecule, adahira ukutod. Okay, so another type of restriction enzyme is the more common one, the endonucleases, which is actually the exact opposite of exo. Kung itong exo, itong pagkakat itong enzyme, itong mga katapusan, itong endonucleases, they cut, so better nga cleave pagkat. So they cut or cleave the DNA, or they cut or cleave within the DNA or RNA molecule. So they cut the internal phosphodiester bonds within a DNA molecule. Do you know what a phosphodiester bond is? I think I mentioned this in when we were discussing the DNA. Phosphodiester bond. Do you still remember this one? Phosphodiester bond. Di ba nasugad? Phosphate. Tapos sugar. Tapos base. Ano? Are you still there?
Opo.
Okay.
Yes. Opo, ma'am.
Okay, now. So the phosphate is connected to sugar, right? The sugar is connected to a base. Let's say, for example, this is a base. Oh, this one is the phosphate, and the phosphate in here. Now, this phosphate is connected to a sugar. It is bonded to a sugar through a phosphodiester bond. As in here, ito nagko-connect from phosphate to sugar. Now, the endonuclease cuts this part here, okay, of a DNA molecule. The endonuclease cuts this part here of a nucleotide base in a DNA molecule. So ginpuputol niya, adan niya puputula the bond that connects phosphate to sugar. Okay, tawag hiton, ang bandha, ang tawag hit niya, phosphodiester bond. Okay. Okay, so the endonucleases can recognize specific base sequence or recognition site or restriction site within DNA or RNA molecule and cleave internal phosphodiester bonds within a DNA molecule. Examples are ECO R1, HIND3, BOM-H1, and others. We will come to that later. Ang mga ano inihira. We will come to that later. Okay, so we go to the classification of restriction endonucleases. So the restriction endonucleases are classified into three. So there are three major classes. What are these? Type 1 restriction enzymes, type 2 restriction enzymes, and type 3 restriction enzymes. Now, the three major classes of restriction on endonucleases are based on the type of sequence recognized, the nature of the cut, and the enzyme structure. Okay? Mag-eriba kasi, type 1, for example, the type 1 restriction enzyme cuts an specific sequence of a DNA molecule. So may adala niya kinikilala nga sequence, ginra-recognize nga sequence. Ibaliwat iton iya klase hiton cut, which we will discuss later. And the enzyme structure. Okay, again, the three major classes of restriction endonucleases are based on the sequence recognized, the nature of the cut, and the enzyme structure. The nature of the cut made in the DNA and the enzyme structure. Please take note of this. Okay? Based on these characteristics, restriction endonucleases were then classified into three: type 1, type 2, and type 3. Okay? Okay na? Can we proceed? Can we proceed? Again, please take down notes. Medyo complicated kasi nga topic. Okay, now, since there are hundreds of restriction enzymes that are already discovered inside cells, or restriction enzymes that are already discovered in cells of many organisms, it is just, I think, proper to name them. So, paano hiragin nga ngaranan? So our next topic is on the nomenclature or naming of restriction enzymes. So meron nga in nomenclature, how are enzymes named? How are enzymes named? In biology, when I think your biology will be on the fourth quarter, but then hit on fourth quarter for this school year. So you will be naming organisms. You will be naming organisms. You will be studying the nomenclature of organisms and itong paggangaran itong mga scientific names of organisms. The topic is actually nomenclature.
There's yellow things coming out of it.
That's not big.
Wait, 1 minute just passed by. I thought you were fast.
One goodness.
No, and
She stopped talking.
Can you turn off the microphone, please? Okay, thank you for that. Thank you. Okay. So again, as what I've said, we'll come to how restriction enzymes are named. Okay. every restriction enzyme would have a specific name which would identify it uniquely. Kasi its enzyme naman is unique. So since its enzyme is unique, they are given names. So the first three letters, please take note of this because I will include, this is, may add ako kasi ganyan mga summative test and I have included this in the summative. So the first three letters in italics indicate the biological source of the enzymes. Again, the first three letters in italics indicate the biological source of the enzymes. First letter. If it is echo R1, so the first letter refers to the generic name. It refers to the genus name or the generic name of the organism. Okay? Kung diin hibibil nga ang restriction enzyme, let's say for example, aada hiya ha E. coli or Escherichia coli. Escherichia coli is a common bacterium used in genetic engineering. So the E. coli. So in naming the restriction enzyme, the first letter, let's take for example the name of the enzyme, the restriction enzyme is echo R1. Echo R1. So the first letter E come from the E. The first letter comes from the generic name of the bacteria or the bacterium. So the generic name of the bacterium. So E for Escherichia coli. So the first letter of the bacterium. The first letter of the generic name of the bacterium rather. So E. Okay. So the second and third being the first two letters of the species name. So ito masunod ko, CO, which actually came from the specific name of the bacterium. So E, co. This is the generic name or the genus name, while this one is the species name or specific name of the bacterium. So at imo kukuhaon hiton generic name is only the first letter. It imo kukuhaon naman hiton specific name will only be the first two letters. Second and the third being the first two letters. First and second letters of the specific name. Okay na? For this one, so H, I, N. So hin. For Diplococcus pneumoniae, D, P, N. Are you following?
Yes.
Yes.
Very good. Very good. So please take note ha. Itong pagngangaran, so itong ngaran, itong restriction enzyme, the first letter of the name of the restriction enzyme comes from the generic name, first letter of the generic name. So kung Escherichia, which is the generic name of Escherichia coli, so E. For Haemophilus influenzae, it is H. For Diplococcus pneumoniae, it is D. Okay, now. Second and third being the first two letters of the specific name. So the next is the second and third. The inihirakuha nga second and third letters, of the restriction enzyme or the name of the restriction enzyme. Dida hiyakuhaan species name or a specific name of the organism. CO for Escherichia coli, IN for Haemophilus influenzae, PN for Diplococcus pneumoniae. Okay? I hope you're following. Then comes a letter that identifies the strain of bacteria. Kasi iton bacteria naman, damot hiya nga strains. Parehas iton virus, damot iton hiya nga strains. So, the fourth letter, didi na hiton ngaran, hiton restriction enzyme, actually comes from the strain of bacteria. Okay? The letter that identifies the strain of bacteria, which is written in capital letter, which is written in big letter. So R for strain R in Escherichia coli. So echo R1. Ano inihiyang R? Strain han E. coli. Strain. Ano yung strain? Strain R. Okay? Strain R. Okay. So next. Finally, there is a Roman numeral for the particular enzyme if there are more than one in the strain. Siguntahan strain R, may ada damo nga strains, may ada strain 1, strain 2, 3, 4, and so on. So damo hiya nga strains itong strain R. Kung ano an imo kinuhaan, kung tikang ito niya diin nga enzyme, sigunta didahan strain R1, then you will write EOR1. Kung ang restriction enzyme, tikang hiyahin strain R2, didayan second strain, an R. So itong imo ibubutang, or itong iyangaran, magiging EOR2. Okay? Nakuha? Were you able to get it?
Yes.
Yes.
Opo.
Okay.
Yes.
So we'll have a short exercise for this. Okay. This name the restriction enzyme. This enzyme was discovered in Haemophilus influenza strain D and was the third enzyme discovered in that organism. So what will be the name of the restriction enzyme? You can write your answer directly on the monitor.
Hindi, third.
Okay, Haemophilus influenza strain D and was the third enzyme discovered in that organism. First enzyme discovered, the third enzyme discovered in that organism. So how will you, or what is the name of that restriction enzyme? How will you write it? Okay, so if you are done, you can write your answer directly on the monitor. Pwede mo na lang i-type using text. Didahiton annotation. Just use text.
It's screen po ba?
Opo, oo. Has-screen na la. Has-screen na la i-type. You can use text in the annotation. You can use text. Okay, you started it right. Thank you for that. Okay, and the answer is correct. Thank you for that. Thank you. Very good. So, hin, D3. H for the first letter, IN for the second and the third, then D for the strain, and 3. Kasi third enzyme discovered na hiya in that organism. Thank you for that. Okay. So, okay, thank you. Do you want more? Do you want more exercise? Do you want more exercise?
Yes, ma'am.
Okay. What if? So what will be the name of the what will be the name of, or how will you write the name of the restriction enzyme if the restriction enzyme is, ano, mimiling koan. Enzyme. Ouch. Ano, nahang pa yan. Later na la. I did na ubos ngayon it mga ngaran. Anyway, so anyway, so basta siya na to. At least you already know how to write the name of the restriction enzyme. Or you already know how restriction enzymes are named. Okay, so for restriction, more on restriction enzymes. So restriction enzymes were discovered in bacteria. It was discovered in bacteria. Anong ira gamit ang bacteria? Anong gamit itong restriction enzyme in bacteria? Bacteria use them as a defense mechanism to cut up the DNA of viruses or other bacteria. Defense mechanism. So, ang binubuhat kasi, let's say, for example, the bacteriophage. You know what a bacteriophage is? Is it a bacterium or a virus? Class.
Bacterium.
Bacteriophage. Is it a bacterium or a virus?
Virus.
A virus. Virus, very good. Virus. The virus that attacks or infects bacteria. So itong ira kinakaon itong ira ina-attack yung mga bacteria. So kay bacteria man ang ira target, ang bacteria may adito nira defense mechanism para lutera di magpatay. God is good. So an maatake ang virus that wants to infect them is the restriction enzyme. So, ang mga restriction enzyme, may adahit na niya recognize na certain sequences in the DNA of the virus, tapos gin-uutod-utod ito nira. They're doing this para iton virus diri mag-multiply hasulod itong bacterium. Okay, so defense mechanism nira. So they cut up the DNA of the viruses or other bacteria that want to infect them. So asan gamitan restriction enzyme din ang bacteria. So ini naman gintap inihan scientists nga restriction enzymes that are actually naturally produced in bacteria. So gintap inihan scientists. So, kay na-cut man hirahin ng specific portions of the DNA, specific nucleotide sequences ng the DNA, pwede hiya magamit dito in genetic engineering. Pwede hiya magamit dito ng genetic engineering kung magka-cut dito segment ng DNA molecule. So, hundreds of different restriction enzymes have been isolated. Its restriction enzyme cuts DNA at a specific base sequence, as what I have said just a while ago. For example, EcoR1 cuts DNA at this specific sequence, GAATTC. Remember this, class. Itong EcoR1, since this is very specific, the enzyme is very specific, it only cuts this base sequence, GAATTC. Igre-recognize ito niya. Meaning to say, diri nga ni ito niya makaagi hining nga sequence, it won't cut the DNA. If a DNA doesn't have this base sequence, then waray igkakat hitong EcoR1. That's how specific the enzyme is. Again, it always cuts DNA at a specific base sequence. An example given here is the EcoR1. So itong ikakat niya, itong may adalahin nga base sequence, GAATTC. Okay, hain niya ikakat between G and A, between A and G below. What? Now, the sequence GAATTC appears 3 times in the DNA strand below. As a result, the strand is cut into 4 pieces. GAATTC. 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 Okay. Cut niya. Pagbubulag inihiya, G and A. GAATTC isang magbubulag dito. Okay. As a result, the strand is cut into 4 pieces. Kasi, ang na-identify han EcoR1 dida han DNA molecule tulo. 1, 2, 3. Ayan na, GAATTC. Okay? And it was able to form 4 segmented or 4 fragments of a DNA. 4 DNA fragments. How do they cut? Okay. Restriction enzymes cut in a sticky ends. So this is one way of cutting. Another way of cutting is through blunt ends. Okay. Okay. So GAATTC and iya pag-cut between G and A on top, di din naman han second strand or the strand below between A and G. For this, between C and G and between G and C, tapos ang klase niya to cut is blunt ends. So the other one is sticky ends. So these are actually the 2 types of cuts. So other restriction enzymes cut at different sites. Examples, some examples are listed below. Okay, for this one. Now, for BOM H1, This is the sequence, the specific sequence that it cuts. G, G, A, T, C, C. For HAE3, G, G, C, C. For PST1, C, T, G, C, A, G. For HIND1, G, A. What is this? There in the G, A, N, huh? Please make the necessary correction. There is no such thing as N in a nucleotide base. Okay, so these are actually specific sites or recognition sites where a certain or any specific restriction enzyme cuts. Okay. Okay, that is. So, tawag hini nga ka-cut is sticky ends. Kung dire-diretso la, we call it as blunt end. So the type of cut is blunt end. Kung inihiya nga diretso la. Pero kung zigzag, we call it as sticky ends. Dili gud actually zigzag. Sticky ends have complementary bases so they could join. Para sen kunta ini sugad ya. Ini nak-cut, adi naman ging cut. So kunta ini foreign DNA look at the ako. So look at me. So pag in sticky ends, they have complementary bases so they could join sugad. Iton was this, blunt, kumbaga gintunga lahiya, tunga, tapos pag join, sugad niya. In sticky end, sugad nga kakat, sugad nga kakat, or sugad. Ano ba? Sugad. Madugtong nahiya. Okay? Okay, so if the vector and the gene to be cloned are both cut with the same restriction enzymes, they will both have complementary sticky ends. Now, class, kung ang gin-cut ha restriction enzyme, let's say for example, EcoR1, didihiton usa nga cell, gagamiton gihapon nga restriction enzyme is EcoR1. So if the vector and the gene to be cloned are both cut with the same restriction enzyme, they will both have complementary sticky ends. After cutting, the two DNA samples are mixed. Okay. Now, I have uploaded this in the Google Classroom. And I have also posted this just a while ago, before we started the discussion. I have posted this because this is very important when you do the activity, when you do the restriction activity. So, dinhi ka mo makuha. This will be your source of answers. So, dinhi ka mo makuha. Let's say, for example, the first one, Bacillus ameloliquefaciens H. So the name of the restriction enzyme is BAM. The strand is H, and it is the first strand discovered in that organism. So BAM-H1. Hain hiya makat between G and G on the 5' to 3' direction, and between G and G in the 3' to 5' direction. So, pagkat niya dida sa magbubulag and G here. Pagkat niya dinhi magbubulag here. So, this will now be the result. Okay? Nakukuha niyo? Okay. Tapos, didahiton iyo answer Didahiton iyo answer, lilinyahan it niyo sumasugad hiya. Nakuha.
Opo, opo.
Masugad. Okay, pasugad hiya hiton ha. Didat iyo pag-answer na. Okay. Ha?
Ma'am,
Let's take, yes.
there are
Yes.
students po in the waiting room.
Ah, okay.
Nag-wait po.
Okay, okay,
Kanina
okay.
pa po.
Ah, hala, sorry. Naya, wait la. Kaya siya nakukuha ng kay basta. Nagdi-discuss na ako dire ko na. Anyway, sato na. Ano na to? Aray na. Okay na? Ano, wala pa kayo erase. Ano, ha? Okay. So, ay nakitahan to. Let's say, for example, the second one. For this restriction enzyme, B, from the generic name, GL for the specific name, so BGL, tapos I or I1, I think this is I1. Anyway, so itong pag-cut niya is between A and G, and then here. Kuha. What is this type of cut, class? Sticky or blunt?
Sticky.
Sticky. Very good. Sticky. Okay, so just do the other enzymes. Just practice cutting the other enzymes after the discussion. Now, what do you notice, class? What do you notice in the cleaving sites? What do you notice in the cleaving sites? GGATCC, CCTAGG, AGATCT, TCTAGA. What do you notice here? GGTACC, CCATGG. What do you notice in the sequence?
They're inverse.
Who answered that? Who answered that? Thank you, Anna, and your answer is correct. Now, you will notice kung ano ang sequence dinhi asya hiya didiha strand below. G, G, A, T, C, C. G, G, A, T, C, C. Let's go to the second example. A, G, A, T, C, T. A, G, A, T, C, T. Okay, let's take, for example, this one. G, T, T, A, C. G, T, T, A, C. Now, we call this sequence as palindrome. Asan niya ang sinisinga? Palindromic sequence. Sarap ko nala, ha? This is very palindrome. Single L, double L. Single L.
Single L.
Single L. Okay. So, palindrome. Tawagin niya, hininga bugos, is palindrome. Itong sequence naman, we call it as palindrome sequence. Now, when we say palindromic sequence, itong iya sequence in one strand is just an inversion of the sequence in the other strand. Okay? Kuha. Now, tanan iton hiya nga mga cuts are all palindromes. They are all palindromic sequences, as you noticed in the examples given here. Okay, and makanotice ka mo. A, G, C, T, T on the opposite strand. Kabaliktaran la. A, C, G, C, T, T. Diba kabaliktaran la hiya? Diba mga kabaliktaran la? Now, lahat ng cleavage sites or restriction sites are all palindromes. They are all palindromic sequence. Okay. Okay, so we'll continue. Wala, ano na yun? Yes. Any question?
There's another student po.
Ha? Meron na sulod.
Meron naman po student na nakakasunod.
Rome.
There po nakakasunod. Sorry po.
Anay ha, anay kay wait la, nahang. Okay. Ay. Problema kasi ta computer nahang hiya. Anay ha. Okay. By the way, class. Okay, adipok na. Okay. Now, after the discussion, you can start doing the activity para dire ka mo hingalimot. And I think I will schedule ano yun, nahang patak computer ha, hiton tisulod. We will schedule the summative test this week, I think. When do you wish to have it? Wala nahang hiya. Oh, okay. Ito na, okay na? Okay. So, okay. So we'll proceed. Now, aside from restriction in enzyme, which is a very important tool in genetic engineering, another enzyme that is used is the DNA ligase. DNA ligase is not a restriction enzyme. Itong mga restriction enzyme, itong role itong genetic engineering is simply to cut the DNA sequence. Cut. Now, what about the DNA ligase? What do you think is the role of DNA ligase in genetic engineering, class?
Together fragments of DNA.
Join together fragments of
Okay, very good.
DNA.
Okay, joining one gene onto another. Okay, so hira iton kumbaga hira kun gingkat naman ano gingkat iton purpose iton pagkat para undesired gene nga imo kukuhaan in one cell in a different cell i-insert mo ngadto han usa nga cell. It can be same organism or it can be a different organism. Let's say for example, nagkuha ka gene from a human cell, it transfer mo nga to a bacterium. Let's say for example, then imo kinuha nga desired gene in the human cell is the gene that can produce insulin. Ginagamit mga diabetics. The gene that can produce insulin, transfer mo ito niya nga gene from human cell into bacterium. Ginamit mo pag-cut is the restriction enzyme. Yan na naman, i-insert mo na hiya adto nga gene from the human cell into the plasmid of the bacterium. So into the bacterial cell. Paano mo hiya i-join? Dapat kasi hiya i-join. Dapat kasi glue. Di ka man magamitin glue. So, you will use the DNA ligase. So, it is the ligase's job to join together fragments of newly synthesized DNA to form a seamless strand. So, ipipaste mo na hiya. Paano mo hiya ipipaste? Using the DNA ligase. Okay? So, another is the Alkaline Phosphatases. This is a group of enzymes which remove a phosphate from a variety of substrates like DNA and RNA proteins. Remove a phosphate from a variety of substrates. Okay? So next, we have polymerases. Anong gamit ang polymerases? I think, okay, so we have 3 important polymerases given here. The DNA-dependent DNA polymerase that replicates DNA from DNA. I think we have discussed this in replication. DNA-dependent polymerase. We have the RNA-dependent DNA polymerase or the reverse transcriptase, and the DNA-dependent RNA polymerase that transcribes RNA from DNA. Now, of the 3 mentioned polymerases, the only polymerase that was not discussed is the RNA-dependent Okay, so of the 3, that's what I said, of the 3 polymerases mentioned here, only the RNA-dependent DNA polymerase was not discussed. Now, RNA-dependent DNA polymerase is also called as reverse transcriptase. Reverse transcriptase that transcribes DNA from RNA. Now, RNA is single-stranded, right? Diba? Yeah. So, garon ta. Drawing na lang kita. If this is DNA, so double-stranded hiya itong DNA. Itong RNA is single-stranded. Now, okay la itong DNA because it is double-stranded. It is double-stranded. So, through DNA polymerase, makakahimuhiya in another strand because okay, madam. Now, paano kasi itong what's this? In restriction enzymes, it only cuts 2-stranded nucleic acid. Paano kung itong i-cut RNA which is single-stranded? So itong bubuhaton itong cell, it will use another enzyme called reverse transcriptase. So itong enzyme, magkukopya hiya didahin specific sequence para hiya makahimo in another strand. Tapos itong hiya nga newly synthesized strand nga naghimo reverse transcriptase from an RNA molecule, we call it as the cDNA or complementary DNA. Again, itong cDNA is just a product. This is actually a product of the reverse transcriptase enzyme that copied a certain segment in an RNA molecule. Nakuha niyo? So, itong DNA, okay lang itong DNA kay double-stranded hiya. Okay? Kayang-kaya itong pagkopya itong polymerase. Kayang-kaya niya mag-synthesize in DNA polymerases. In the case of RNA, kay uusaman la iton iya strand, di ba? Uusala iton iya strand, uusala iniya nga strand. So, paano hiya makakahimo hin another strand? Kaya dapat kasi double-stranded. So, paano hiya makakahimo another strand? So, the cell will use the reverse transcriptase enzyme to copy a certain segment of the RNA molecule. So magkokopya hiya. Kokopyahon ito niya, makakahimo hiya hini. And iya na himo is then called as complementary DNA. Meaning to say, ini nga DNA was actually copied from an RNA molecule. Nakuha niyo? Were you able to get it? Okay. Okay, so for the polymerases, there are 3 important polymerases used in genetic engineering. The DNA polymerase, or the DNA-dependent DNA polymerase that is used in DNA replication. The DNA-dependent RNA polymerase that is used in transcription, and the RNA-dependent DNA polymerase that uses reverse transcriptase enzyme to copy the sequence in an RNA molecule. So the reverse transcriptase, or the RNA-dependent DNA polymerase, transcribes DNA from RNA. We call this RNA-dependent DNA polymerase as reverse transcriptase. Okay, so one tool in genetic engineering is, or enzymes. The enzymes, so for the enzymes, we have the polymerases, we have the DNA ligase, and restriction enzymes. They are all needed in genetic engineering. So another tool in genetic engineering are the vectors, or the cloning vectors. Vectors or cloning vectors. The vectors, or the vector is a DNA molecule used as a vehicle to transfer foreign genetic material into another cell. DNA molecule again, a vector. When you say vector or cloning vector, it is a DNA molecule that is used as a vehicle to transfer foreign genetic material into another cell. Let's take plasmid as a vector. So plasmid. So, meron ako drawing. Okay, so we have it here. Plasmid in here. Let's take, for example, this one, a bacterium. Aside from the bacterial chromosome, may ada ito niya circular chromosome, which we call as plasmid. So, plasmids have shorter DNA sequence or DNA molecules or DNA nucleotides as compared to the bacterial DNA. Now, go back. So the plasmids are small circular pieces of DNA that are not part of the bacterial genome but are capable of self-replication. The plasmids are used as vectors to transport genes between microorganisms. Once the gene of interest has been amplified with PCR, the gene and plasmid are cut by restriction enzymes and ligated together. The resulting combination is known as recombinant DNA. We'll go back to this illustration. So, in genetic engineering, they don't use the bacterial DNA, but the plasmid. Iniya nga plasmid, di ba? Circular DNA inihiya. It-aan nga DNA mas guti as compared to the bacterial DNA. This is where scientists or technologists undergo genetic engineering. This is where scientists or technologists enter the desired trait from, or that they get from, other cells. Dinhi ibubutang. Let's take for example, the gene for human insulin. So, ang human insulin, di niya kuhaahin human cell. So, adto nga kinuha nga human insulin or insulin from a human cell, i-insert niya didi ha plasmid. Okay? Like this one.
Mama,
So,
may
ikakat.
It's waiting room.
Okay. Thank you. So, ikakat ito niya nga plasmid para mahi-insert ang foreign gene. So, meron nga foreign gene, it is a gene that was taken from other cells. Okay? Or a gene that was taken from the cells of other organisms. Okay? So, ikakat ito niya through a restriction enzyme para hiya i-insert. Okay? Yes. Inihiya, i-insert dida, i-insert. Dida, sugad. Inihiya ma-insert nga di. We call this type of cut as sticky ends. So inihiya, enlarge la iniha, tikan inihiya didi. So inihiya, ma-stick dida, para hiya. Okay, ma-stick hiya dida, para hiya mag-join. Ini, adi naman. Can you follow? Okay. So again, restriction enzyme is used for cutting or for cleaving the foreign gene and the plasmid. The DNA ligase's role is to paste it or to join them together. Okay, so aside from plasmid, we have the viral vectors. Okay, so vectors coming from viruses. Just like plasmids, they are also used for the delivery of genetic material into cells. Okay? Foreign gene, gin-modify na hiya, gin-genetically engineer na hiya. So may add na hiton dida desired gene, magamit hin virus, then the virus will inject its DNA into the bacterium. The DNA with the foreign gene will then be incorporated into the plasmid of the bacterium. Okay. Okay. So another vector is the Cosmid. There are actually hybrids between plasmid and Feds lambda. The Feds lambda actually come from the bacteriophage, which is a virus. Okay, so the Feds lambda can carry large fragments of DNA. So designed to clone large fragments of DNA and to grow their DNA as a virus or a plasmid. Okay. So this is not a Cosmid. Mid from plasmid. Cos from the alphaphage. Alphaphage naman from the phage lambda, from the bacteriophage. So this is the cos site. So organic cos site. The specific recognition site where foreign DNA is inserted. Okay, so aside from Cosmid, we also have artificial chromosomes. The artificial chromosome is used to clone relatively large fragments of DNA. So we have two examples of artificial chromosomes. We have the bacterial artificial chromosome or the BAC. And we have the yeast artificial chromosome. So kung iton imo i-insert dako hinduro nga fragment of a DNA para hiya ma-accommodate, para hiya eksakto, para hiya ma-accommodate, para iton nga large DNA fragment ma-accommodate into a certain DNA. So it imo gagamiton will be the yeast artificial chromosome. The specific yeast used for this is Saccharomyces cerevisiae or the baker's yeast. So there are generally engineered chromosomes derived from a DNA of the yeast, which is then ligated into a bacterial plasmid. So ibubutang mo laanay an undesired gene, let's say for example from the human cell, ibubutang mo nga diha yeast DNA, tapos from the yeast DNA into the plasmid. Into the bacterial plasmid. And bacterial plasmid, hiya na ang mag-a-amplify. When we say amplify, hiya na ang magpapadamo ang desired gene. Let's say, for example, insulin kunta. Kaya mo magdamo ang insulin. So, ibubutang mo hiya nga daha plasmid. Tapos, when the bacterium multiplies, magmumultiply gihapon ang plasmid, carrying with it the foreign gene. So from 1, it will become 2, it will become tens of bacteria, and then tens will multiply, it will divide through binary fission until such time that it will become thousands of it. So, ang thousands, makaka-produce na hinda mo nga insulin. Okay, so I think I have, yes? Yes?
Damo na naman po na disconnect.
Okay, thank you for that. Okay, thank you. Kaya naghihininay kasi kuan basta asya nga pwede atong klase aga nala kay basta ti kakadto nat oras nagkakamata na kasi nagpipinan uyas nalo yan at internet pwede atong klase aga nala kay paro kakulo malilisang na akay wala na makaka-connect mahinay na ngayon ang internet connection. Okay, anyway. Okay, so I don't know if I have posted this in the Google Classroom. Parang, I can't remember. So here is the difference between a plasmid and a cosmid. Just read it silently. Kung kay ano nga hybrid hiya, this is a combination of a plasmid and a virus. And virus din is a bacteriophage for the cosmid. Okay, now look at the plasmid. A plasmid can accommodate only a DNA piece of up to 15 kilobases. So meron nga kilobase, 1 kilobase is equivalent to 1,000 nucleotide bases. So a plasmid can only accommodate 15,000 nucleotide bases or 15 kilobase, while a cosmid can accommodate up to 45,000 nucleotide bases. So, mas madamo ang iya makukuan, mas madamo ang iya ma-accommodate. Okay. So okay, transformation frequency is low, while in Cosmid, transformation frequency is high. Later, we will explain what transformation is. Okay, so these are examples of plasmids, while this is for cosmids. No. May ada ko din. Okay, so another tool in genetic engineering is polymerase chain reaction. I don't know if you are familiar with this machine. Are you familiar with RT-PCR? Class, are you still there?
Yes.
Okay, are you familiar with RT-PCR?
Yes.
So, ano mo't RT-PCR? Panitong hiya. RT from reverse transcription polymerase chain reaction. Or negative from COVID. So itong ginagamit ng machine is RT-PCR. Masing ka naman, ma'am, kay ano nga RT? Kay ano nga PCR nala polymerase chain reaction? Kaya ano nga may reverse transcription? Kaya ano nga may RT? Alam mo, RT yung PCR. So kaya anong RT? Kaya anong RT class? Yeah, sige daw. Anybody? Kaya ano nga may RT? RT comes from reverse transcriptase. Because, sige, ako nala, pakian, ako't mabaton. Ano? So, kung kay ano nga may reverse transcriptase, because COVID virus uses RNA as its genetic material. Itong bacteriophage kasi, ito na ginagamit na genetic material is DNA. This, I remember when there was a question in one of the summative tests before, pakiana dito, what genetic material is found in the bacteriophage? And iyo naman answer, common answer, both DNA and RNA. Dili pwede, dapat usala itong genetic material. It is either RNA or DNA. Most cells use most cells use DNA as their genetic material. Pero there are certain viruses that use RNA as their genetic material. COVID virus is an example of this. Its genetic material is not DNA but RNA. Para naman hiya magdamo, pag-amplify, pagpadamo, magamit hiyahin PCR, the polymerase chain reaction. Para naman hiya mapadamo, magamit hiyahin restriction enzyme. Restriction enzyme, magamit hiyahin RT, magamit hiyahin reverse transcriptase. Okay, magamit hiyahin reverse transcriptase. Kasi the COVID virus, again, has RNA. It uses RNA. So para mapadamo ito nga RNA, para mapadamo ito nga what's this? Para mapadamo, amplify ito nga molecule, it will use the reverse transcriptase. Okay, so RT-PCR is reverse transcriptase polymerase chain reaction. Anyway, so polymerase chain reaction or the PCR is one of the tools used in genetic engineering. This is actually a machine. So it iya hinigamit is to amplify para mapadamo itong molecule, itong DNA molecule. Again, kay ano nga may arte, kay waray itong COVID, waray niya DNA. Para makain mo yan DNA, magamitin niya in reverse transcriptase, which is an enzyme. Okay, so polymerase. So polymerase chain reaction is sometimes called molecular photocopying. Molecular photocopying. Parang nagpapa-xerox kita, nagpapa-photocopy. Itong usang original copy, pwede mo itong hiya ma-reproduce many times. Pwede mo itong hiya ma-reproduce 100 times, thousands, or even millions. Okay? So, para ya mapadamo, itong DNA polymerase chain reaction PCR is used. Okay? So what is PCR used for? Hain hiya gagamita. Once amplified, kunta napadamo na hiya. Using PCR, so barangan amplify pagpadamo. So the DNA produced by PCR can be used, one, for laboratory procedures. Okay? For laboratory procedures, kung ta may mo gini-studyhan na certain disease, or may mo gini-studyhan para kung tahiton pag-identify hiton kung positive ka, kung may ada ka hiton nga virus, kung positive ka hiton nga virus, dapat i-amplify hiya. Padadamoon hiya through PCR. So the amplified DNA can be used in laboratory procedures. Another, it can be used in DNA fingerprinting. DNA fingerprinting, you know what DNA fingerprinting is? Fingerprinting is a clinical technique. Ito nga, pag-identify itong anot sequence itong imo DNA. Anot sequence itong DNA. Itong ginagamit ini, itong fingerprinting ginagamit itong pag-identify kung ano itong kung hino di ba ginagamit itong kung hino ginagamit kung hino itong imo nanay, hino itong imo tatay, or imo mga kabugtuan. Okay. So igbabasahon itong sequence itong imo DNA. We call that process as DNA fingerprinting. So kung ta may ada dida patay tapos or krimen, for example, krimen. So may ada dida blood sample. So paano hiya ma-identify nga ini hiya as siya an murderer? So i-fingerprint iton hiya. Kikitaon dida blood sample and DNA sequence. Through that DNA sequence, ma-identify ang tawo nga may ada parihas hiton nga DNA sequence. We call that process as DNA fingerprinting. Now, PCR is used also for detection of bacteria, viruses. Okay, di ba? Detection of bacteria, detection of viruses. Asya nga ginagamit inihiya, dida iton COVID test. Okay.
Ma'am, may na-disconnect at the waiting room.
Damo, okay, nga na disconnect anay. Kinaudong paliwak ito na anyway. Ay naman ito. Hala, papaluti yan. Wait la. Ay naman ito. You are. Okay. Okay, so fine na kita. So what is PCR used for? One is in laboratory procedures, DNA fingerprinting, detection of bacteria or viruses, and the last one is for the diagnosis of genetic disorders. Ano ba yung genetic disorder? May ada ka ba? Marfan syndrome, may ada ka ba sickle cell anemia, may ada ka ba Down syndrome. So to diagnose or in the diagnosis of these certain disorders, PCR is used. Okay, so for the mechanism of the PCR, just use you can search on this through YouTube. Okay, so this is how it is, how the molecule is amplified. Papadamo here from one DNA, madadamo here. Can become millions. Okay, so another is gel electrophoresis, another tool. Ini yung mga machine na inihiya. There na yung mga substances found inside the cell, like the restriction enzymes, like the mga polymerases, the vectors. These are already machines. The PCR and the gel electrophoresis. Ito na gel electrophoresis naman, it gamitin niya kung taging cut na hiya na ginutod-utod na hiya. Now, the gel electrophoresis is now used to separate the DNA fragments. Okay, the DNA fragments or the RNA fragments. Okay, so this is or an illustration of a gel electrophoresis. Okay, so another is the prokaryotic host. The most common prokaryotic host used in genetic engineering is the E. coli. The E. coli is used as a vector. It is also used as a storage of desired genes. Masing ka naman, ma'am, why of the so many bacteria in the environment, of the so many viruses that we can use in genetic engineering, why E. coli? The answer is, under a suitable environment, the number of E. coli can double every 20 minutes. So makusugit iya, multiplication, makusugit iya pagdamo. As the bacteria multiply, their plasmids, along with foreign DNA, also multiply to produce millions of copies, referred to as a colony or in a short, or clone. Okay. Aside from being inexpensive, prokaryotic, the E. coli easily multiplies. So another, aside from the prokaryotic host, another tool in genetic engineering is eukaryotic host. Okay, the most commonly used eukaryotic organism is, what do you think?
Yeast.
Yes, very good. The Saccharomyces cerevisiae. So in bacteria, the most common host is the E. coli. In eukaryotes, the most common is the yeast or Saccharomyces cerevisiae. Okay, so we go to transformation and transduction. So transformation, numerical transformation in biology, it refers to the direct uptake of incorporation of DNA from their surroundings. Drawing nga kita. Let's take for example. This is the bacterium. Adi an iya plasmid. Plasmid ini iya plasmid. Adi an iya bacterial cell. Bacterial cell, bacterial chromosome. Okay, adi mga plasmids. So, adi an foreign gene. Foreign gene. Sigunta adi an foreign gene. Inihiya an foreign gene. Now, in transformation, okay. So in transformation, inihiya, there will be a direct uptake of this DNA into the cell, into the plasmid. While in transduction, the foreign gene is transferred from one bacterium to another through a bacteriophage. Now, in transformation, inikusa nga masulod na da. In transduction, iton magdadara hiton foreign gene is a virus. Ano? So, ini, let's take for example, may ada dida virus iton hiya dida virus. I-inject niya ini nga DNA into here. I-inject la niya. Diri masulod iton virus. Diri masulod iton bacteriophage. It will only inject the DNA with the desired gene. Let's say, for example, may adat niya dida human insulin nga gene. So, i-insert dida niya and this will then be incorporated into the plasmid. Okay. Ibubutang itong hiya dida. Okay, so that is the difference between transformation and transduction. Okay, identifying transgenic organisms. Not all cells will take up DNA during transformation. Therefore, it is essential to identify the cells that undergo a transformation and those that have not. Generally, plasmids carry genes for antibiotic resistance, and transgenic cells can be selected based on the expression of those genes and their ability to grow on media containing that antibiotic. Alternative methods of selection. Okay, so how will you identify if the organism has a foreign gene in it? Let's say, for example, dida. So, didahin usa nga kuan iton, usa nga ano ini, medium. So, nadidaan mga damo nga mga bakterya, mga bakterya dida, damo dida mga bakterya. Nakada naman ang mga plasmid, dida mga plasmid. Basta da may mga plasmid dida. Now, question is, how will you know kay diri itong hiratanan nga bakterya ma-absorb foreign DNA through transformation? How can you identify that these bacteria are transgenic? How can you identify that these bacteria have foreign gene inserted into them? Now, itong scientist or itong technologist, magamit yan antibiotic. Most often, itong ginagamit nga antibiotic is ampicillin. So, inihira nga mga bakterya i-introduce hirahin ampicillin. So bubutangan hirahin mga ampicillin. Okay. Iton hiya nga may ampicillin, dida iton hiya nabutangan hin pharyngean. Ano, nabutangan ito na para pag-identify nga iton hiya nga bacteria may ada pharyngean, dapat iton hiya nga bacteria may ada liwat ampicillin. Okay? Now, pag-identify naman kung hain dida iton may pharyngean, hain dida iton waray, nga during harvest time, ini nga medium, ini dida nga ada iton medium, iton binubutangan hini nga mga culture medium actually. Ito niya nga binubutangan hini nga mga bacteria. Iton medium, it actually supply food to the living bacteria, to the living organisms. Now, bubutangan niya hin ampicillin. Butangan niya nga medium hin ampicillin. Itwaray ampicillin, mamamatay. Sigunta ini waray ampicillin, so mamamatay hiya. Mamamatay. Meaning to say, waray niya pharyngean. So ito makaka-survive are those with ampicillin antibiotic in them. And may ada ampicillin antibiotic are those bacteria with pharyngean. So ma-identify mo, mainihiraan may ada pharyngean because they are resistant to ampicillin. Kaya ano nga naging resistant hira? Kaya gimbutangan hira mismo, gin-introduce hira in ampicillin nga antibiotic. Okay? So I hope you were able to get it. Okay? So for a summary of the lesson, in the tools of genetic engineering, the scientists or the technologists do not use X, common tools in the kitchen. They don't use scissors, but they use enzymes to cut certain or specific segments of DNA. They use ligase to join the fragments in the DNA molecule. They use also what else? Vectors for storage of large fragments of DNA. And aside from that, they also use PCR for amplifying molecules. They use the gel electrophoresis for separating fragments. They use the prokaryotic host and the eukaryotic hosts to modify and improve certain cells or organisms. I hope you learned something from today's discussion. Did you? Did you learn something?
Yes.
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 Okay. So, to test if you have learned something from the discussion, do at once the extraction enzyme activity. Okay, so in the next, what's this? In the next distribution of modules, you will submit your output to the barangay distributor. Okay, so the first activity will be submitted on Monday. The hard copy, hard copy. Do not submit the soft copy because I will find it hard to check. Okay, so submit the hard copy of the activity to the teacher distributor in your barangay on Monday. Okay. That will be our first activity for the third quarter. Now, what about the summative test? When do you wish to have it? By the way, our 3 questions before we go to the summative test schedule. You are free to ask questions. I might upload a video for this topic in the Google Classroom.
Full module topic.
Ano to?
For the whole module.
Module 1 la. Oo, module 1. Itong summative test, module 1 la. Work on restriction enzyme for now. Ayaw anay kabuhan an tanan module 2, module 3. Ayaw la anay. Mo overwhelm ka mo hin mga trabahoon. You work one at a time. So the activity that will be collected on Monday will be the first activity, the restriction enzyme activity. And hala, ano pa nakadan module 1?
Essays.
Essay. Duhala ang module 1 nga required output. Duhala ang required output.
Yeah.
Duhala. Okay. So kung duhala, so essay and the restriction enzyme activity. Now, pwede na kamo diri magkuan, pwede na kamo diri mag-submit han mga other activities in the module. Iton iyo isa-submit, pwede la adto nga required activities. Required activities. Kaya siya iton akon graded and recorded activities. The rest of the activities are recorded but not graded. Again, recorded but not graded. Kaya ito nira mga formative ko anda iton hiya. O hira mga formative assessments. So iton akon igra-record, iton graded and recorded are the required outputs. So you don't have to pass other activities in the module. Pwede na la iton hira nga activities in the module, pwede na la trabaho niyo iyo balay, and then self-check na la kung just refer to the answer key at the end of the module. Kung nakaasya ka mo. Pag-test iton kun iyo na hibaruan in the module. But you don't have to pass it. You don't have to submit it. Basta't akon la gin aaro nga you will pass will be the required outputs that I have listed. Okay na? Okay, iba na submit lagi hapon hin ko na submit lagi hapon hin mga ano ito, other activities in the module. Mada kon durot iyo trabahoon, damo bayat na activities dida. So ginibanan ko niyo trabahoon asa nga nag-identify la akin mga pipirahoy lang activities that you will submit. Ano? Okay la? Okay la kung i-require takpa mo han iba pang activities in the module.
Okay
aside
la.
from the required. Okay, so sige, sarabot ta ha. Itong igpapasla ha. Required outputs that I have listed. Ano, ang ibang activities in the module, pwede na lang niyo trabaho na iyo balay, and then self-check na la, but you don't have to submit it. Okay? Para di magdamot iyo trabahoon. Okay? Okay na. Okay, so i-restore yan naman kita yan na. Anay. Paki turn on hiton iyo paki turn on hiton camera kay isa-submit ko ini nga picture hakuan. Isa-submit ko nga picture nga hiton amon head. Paki please turn on the camera. Thank you for that. Thank you, Jocelyn. Okay. Alexi, paki turn on it yung camera.
Diri na gana.
Diri na gana. Okay, sige. Kaya ikukuhaan ko ini ha. Ano pa daw. I will take a picture and then submit it to my boss.
Nag-look up ang
1, 2, 3. Okay. So, anay ha. Anay, makuha mga picture. Okay, so we'll now discuss the schedule of the summative test. When do you wish to have it? Aman, Alexi, naman. Okay, sige. When do you wish to have the summative test, our first summative test in the third quarter?
Next week.
Next week? Nangalimtan nat niyo. Meron na kamuhi barot pagngaran ni Transduction enzyme. Okay, Friday na buwas uugad ngayon next week. So, when? Next week? When?
Tuesday.
Sunday.
Monday submit.
Monday. Ayaw, wait, Monday kay. Monday, basta hit ka mo. Oo, para sa hit Monday. Oo, ayaw hit Monday kay. Distribution liwat na mo didi barangay.
On Tuesday morning kay Maloy.
Oy, morning tala. Kay lilisang liwat akin dir inak nakaka. Pero kakulop.
Ma'am, Google Classroom lang.
Opo, Google Classroom. Opo. O ano di karuyag? Zoom or Google Classroom?
Google Classroom lang, ma'am.
Okay, sige. At iba, ano di karuyag? Zoom or Google Classroom? Kay Zoom nga ni.
Google Classroom.
Google Classroom.
Google Classroom, ma'am.
Classroom.
Okay. Sige. Again, ano adton schedule?
Tuesday
morning.
Tuesday morning. Okay. Now, kung diri ako mo-available, kay parang may ada again i-restoryahan na activity, kay may tiabot na bisita school. Kung diri ako available itong Tuesday, I'll just post it in our GC, ha? So, kita-kita lang itong GC, ha? Pag may time. Okay? So, the schedule will be morning. Okay, sige. Turn on the camera again for the final attendance. Alam mo, check pa ngayon kan attendance, ano? Ano sige daw, kay iba, waray ko na pagka-record. Basta nagragrado ako, kinikitaan ko lawat inihinot na attend. We'll take Mango. Say present once your name is called. Zion. Araba, Mark.
Present.
Chad.
Present.
Bantiles, John David. Justin Lay.
Present.
Lance. Dick Juno. Ernst Hardillo. The fourth, Malinaw. Christian Andre.
Present.
Alexi.
Present.
Jade Ugale. Carl Vincent. Yeshiel Seso. San Emmanuel Solayaw.
Present.
Marie Vincent. Mary Vincent. Erica.
Present.
Lorraine Abobo. Zoe Afable.
Present.
Gail Alde. Anna Gia.
Present.
Raven Amoyo. Raven. Joyce. Julia Apura. Julia.
Present.
Nash Astakaan. Nash.
Present.
Angeline. Angeline Colima.
Present.
John Antoinette Dator.
Present.
Krishna Jamel is excused. Ghislaine Jolola. Ghislaine.
Present po. Present.
Aliana. Aliana.
Present, ma'am.
Jillian.
Present.
Julia Guevara.
Present po.
Antoinette Labado. Antoinette. Makshan Amara Sigan. Heart.
Present. Present.
Debbie. Debbie Annette.
Present po.
Angel Monroy. Angel. Isabella.
Present, ma'am.
Francesca Promeda. Francesca. Jaslyn Faith. Uno. Riz Sakmar.
Present po.
Angelique Suwal. Okay. We go to section Santol. Joshua Abusejo. Joshua. John Joseph.
Present.
John Edward Are. John Edward. Mark Kenneth. Mark Kenneth Bulanadi. Earl Joseph Catalo.
Present.
Earl Dilantar. Earl. Andre Labro. Renz Lilia.
Present. Present.
Si Andre present. Andre. Is Andre present? Andre Labro. Renz. What about Renz? Renz and present
Present.
kanina. Okay, very good. Hans Lomar. Rosalejos. Raydel. Samson.
Present.
Okay, very good. John Ray Trabaco.
Present, ma'am.
Okay. Princess Abelia. Kera Adamas.
Present.
Francine Alaya.
Present.
Hannah Amos.
Present.
Denise Apura. Josel Pay Banario. Andrea Beato. Miska Cepeda. Kiana Cesista.
Present po. Present.
Jazzy Ray Toldolea. Marian Dulfo. Marian. Cristil Factor. Denisa Fuentes. Hyacinth Galupe. Hyacinth. Tricia Gapud. Angel Garcia. Angel. Liana Gura.
Present.
Hazel Hilaria. Hazel. Shira. Kirsty. Charnet Latoha. Charnet. Giselle Mendoza. Jahara Montes.
Present.
Jazel Padilla. Isaiah Pagayanan.
Present po.
Allison May. Allison May. Yes, Ninia. Ninia
Present.
Ramirez. Cherise. Okay. Cherise Rijang. Grace Love. Sabido.
Present, ma'am.
Okay, very
Present po.
good. Ella.
Present po.
Okay, sige. Okay, so see you on Tuesday morning for the summative test. Just review. And I might, no. I will upload a video in the Google Classroom. Okay, I hope you learned something today. Thank you for bearing with the internet. Thank you for bearing with me. Sorry kakulop balit ta ugod na lisang akong kaywari ak makagkuan. Naghina talagaan kuan, naghina talagaan internet connection. Haiyo, dire na ako net internet. Dire na hinay.
Opo.
Good for you. Na akong kakulop pati link, pati ang Zoom.
Excuse me, ma'am.
Yes.
Ma'am,
Ano?
na-disconnect ka siya. Warak pa ako anhan attendance.
Antoinette.
Yes, ma'am.
Okay. Sige. Gra-record na lang today. Okay, thank you for that. Thank you. I-restorya daw kita handiri pakumpletohan quarter 3, quarter 2 nga outputs. Manahatag na ba yan card? It iyo advisor. Pwede, pag-submit ka mo, may ada pa mga kulang, you can still submit, even if it is late. It is better late than never. Submit lang ka mo. Kaya ihahatag ko na lahat ko. Once completed na, ihahatag ko na, isasubmit ko na lahat na to yung advisor. Bangin naman lang makalanat hit panhatag hit card. Naghatag na hin card ma'am, an advisor. Waray pa.
No.
Ginuhulat pa siguro iyo pag-submit. Asya nga waray pa igahatag it iyo card. Sige, submit na ka mo ha. Submit na ka mo. Okay, sige. So, thank you. And good morning once again. Thank you. Bye.
Good morning. Goodbye.
Thank you po, ma'am.
Okay.
Thank you po, ma'am.
Bye-bye.
Goodbye, ma'am.
Bye. Goodbye.
Bye, ma'am.
Bye, ma'am.
Goodbye.
Bye.
Okay, bye-bye.
Bye-bye.